Osh, welcome to Digital Davos. You told me a lot about your company, and it's a great company, and I cannot explain it as good as you can do. So when you, in short, describe, you put a living cell on a silicon chip, and this chip is then enabled to basically bound proteins on it, antibodies, and it is basically like smelling, that you can detect explosives, that you can detect also diseases. There's a wide variety of implementation, and it's far behind right now for research. You have already the capability to produce these chips and contacts with the Fraunhofer Institute. You took the technology from ETH. So please describe, in short, your company, and we start with question and answer.
Thank you very much, Carl, for having me. I really appreciate to be in Davos today. So our company is called Koniku. We are based in San Francisco, so just outside of San Francisco. What the technology is basically a breathalyzer for disease. Imagine if you had a smell camera that is in your bathroom, that you can breathe on it, and it would essentially diagnose your disease. So in a longitudinal way, every morning you go into the bathroom, you breathe, you do your number 1 and your number 2, and there is a ton of data you give away in the bathroom. We collect all this data, we analyze it, and we can tell you how your health is going. That's essentially what we have developed. A smell camera or a breathalyzer for disease, right? And the technology that is on the back of it is actually quite interesting. So what we have done is we have taken biological cells, so either brain cells, astrocytes, or HEK cells, and we genetically modify them to express the same proteins that you would have in a dog's nose, in a mouse nose, or in a human nose. And we merge these cells with silicon. Okay, so these cells are genetically modified that they breathe. So it's like a cyborg, the first beginnings of a cyborg, if you will. And we analyze that data. The chip has a Wi-Fi or 4G chip in it, so that you can connect to it anywhere you want. So it's essentially an IoT play as well. That you have these devices in every home on this planet. And we can have a population data in terms of how individual healthcare or how individual health is going. In fact, I like to say that Koniku would like to manage health, not disease, so we can spot things way before things go awry.
Thank you. So, some questions about your technology.
Please.
So, you have a chip, and you bound the cell on the chip. Is it only one cell that you bound on it, or multiple cells? And how is the cell bound on the chip?
So what we have done is, it's actually an array, right? So, so it's like a matrix. So you have one population of cells expressing a receptor that responds to only one chemical compound. You have another population that responds to a different chemical compound. So to back up a little bit, why this is important for human health, for instance. So most people know that every time you breathe, out of your lungs, you have carbon dioxide coming out, okay? What a lot of people might not realize is that a large proportion of the gas that is coming out from your lungs is carbon dioxide, but there are also nearly 3,000 other compounds that are in trace amounts. So things like acetone, things like just many other different kind of compounds. And the literature on this is actually quite solid. So over the last 40 years, we have done a lot of research, not us as the academic community, has done a lot of research that has shown that, for instance, to detect lung cancer, your lungs will produce 11 different new volatile organic compounds, they are called. Will produce 11 different new volatile organic compounds, or existing ones in different concentrations. The same thing for liver cirrhosis and so on and so forth. So our chip has multiple channels to be able to capture all of these different compounds, so that when you have an illness, for instance, and there is a change in the concentration or in production of a new chemical compound, we can detect that and have a fingerprint of a disease, if you will. Okay, so it's a chip. It has different locations of different cells that are expressing different proteins, and then we capture that. We analyze that data because you're collecting it from many different locations. You can smoothen that data or average out the data to get differences, if there are differences in populations based on genetics, based on environment, based on what have you. We can easily iron out those data sets. So the cells are in a cassette, if you will. There's a consumable piece. There is a base device that reads the data, and then there is the electronics that has a big IoT data plane behind it.
It seems really incredible. So I think a lot of people will not believe you when you say this. So let's go how the chip interacts with the human cell, which you modify. So I assume the receptors are expressed by the cell wall from the cell after basically normal replication. But so how do you really measure it? Do you measure like changes in the cell membrane, like electricity currency changes, color changes, you have an indicator? So how you measure it really, and how the measurement is done from the chip to the human cell?
So yeah, that is true. When we usually tell people that what we're doing, the reaction is, wow, that sounds like science fiction, until we bring them to our lab and they see the working device, and people are usually shocked. I guess this is what Davos is about, that you come across really very things that are on the forefront. So in terms of how how we get the cells itself to respond and how that works, I think it's very useful to go back to, let's take example of the dog olfaction. Yes, how does the dog olfaction work? So as you guys are here in this room, you are recording the signature, the smell signature of this room, if you will. Now, how is that happening? Smell is a very interesting sensor, because when you detect the smell of something, you are actually detecting particles. Yes, you're detecting actual particles of the thing that you're smelling, which gives a new reading to when you go to the bathroom, yes? Yes. But you're actually having tiny particles of whatever causes that smell going into your nose, okay? Now, when this particle goes into your nose, yes, it comes across a protein, yes? Think of a protein as a string, like just a string that is folded in three-dimensional space. And the particle of the smell is like a tiny marble ball, okay? Now, okay, or maybe a tiny balloon, because marble balls are a little bit heavy, so they don't float very well in the air. So like a tiny, imagine a tiny balloon, okay? And it's bouncing around in 3D space, via what we call a Brownian motion, if you want. So it's just bouncing around randomly. And then when you make this breathing, when you're breathing, the action of your breathing pulls in this particle, and then it goes into your nose, yes? And then it binds to this the three-dimensional string that is on the surface of the cell, captures this little ball of smell, if you will. That action of the protein binding to this tiny ball causes a chain reaction inside that cell, okay? That chain reaction is what you call a pathway. Yes, so we have this in all the trillions of cells in our body. There are different pathways, okay? Now, in the case of the nose, you have what you call an olfactory sensory neuron, an OSN. Now, the binding of that particle to the protein causes the cell to generate what you call an action potential. It's essentially an electrical signal, okay? Now, this electrical signal now goes to a small processing center called the glomeruli. Yes, so the glomeruli. So it's like a ball. It's like a little ball of cells, like a processing center, if you will. Now, this center analyzes the signal that is coming out of this cell, and it goes to what you call a mitral cell. And this mitral cell is what projects directly to your brain. And then, if you imagine this electrical signal just ping in your brain, then your brain does the processing and says, I smell bacon, or I smell chicken, or I smell whatever. Yes? So that's essentially how it works. That exact process, we have transferred that process to a chip using synthetic biology. I like to make the joke that The age that synthetic biology is going to rot on our species is going to be unprecedented. If you think the internet age and the age of electrons was fun, wait until the age of the DNA. And in fact, we just published a book, I and 25 other technologists, 2 weeks ago. Jane Metcalfe, the founder of Wired, she has started a new magazine based on this, and I predict in a few years when we are going to do this in Davos again, it's going to be all the rage, but that's a separate point. So, using synthetic biology, we modify these cells to do essentially the same thing that I've just explained on how the brain works, okay? So what do? We take first a stem cell, yes, so a cell that can essentially be modified into any other kind of cell, and we import new genetic material into the cell. And that same pathway that exists inside the nose of the human being, we recreate that same pathway, okay? So you have a protein, the string that I talked about earlier, that responds to a specific compound, okay? Once we get the protein that responds to a specific compound, we force a cell to express that protein, and we create a pathway that responds up to 200 times faster than what exists in your nose. So essentially, evolution is unintelligent, yes? What evolution has is time. So if you have 20 billion years or 2 billion years to evolve, I mean, you can really do a lot. But in this case, we have machine learning, we have computers, so we can do this evolution within 3 months to make a cell that responds up to 200 times faster and better than what exists in nature. So we now take this cell and we put it inside a substrate, yes, that has a microfluidic framework inside, that has essentially cell juice, if you will, and then this keeps the cells alive. So this, the chip side is a consumable, yes, that you have to replace on a monthly basis or on a yearly basis at regular intervals, but that's a business decision. And there's a base unit that reads the signals that are coming from the cell. Remember I told you earlier that the cell responds in your nose by electrical signals? In our case, we have genetically modified the cells to light up every time they get a particle from the outside. And we read these signals with an FPGA or our own chipset. We analyze these signals, and then we send this signal to a computer, we send it to a smartphone or wherever you want.
Thank you. So it's really interesting. Normally, there is an old test, it's called a ELISA test in medicine. There you have just like receptors, and you bound them on just a flat surface, and of course, the protein structure changes. Do you expect, as you hold now, the receptors in the native form, which don't change, that you will see in a medical application, that you will see that bounding from antibodies, where normally tests were not doing it, and we discover much more antibodies and therefore much earlier disease detections when we would use the cells and combine this medical testing?
So the difference, so the ELISA test that you mentioned earlier, it uses antibodies, right? And it binds to specific areas of the protein, in a protein that is either in its natural state or a state that is compromised. How that protein behaves in an ELISA test versus how it behaves in vivo is a little bit different. For the people that are not into biotech here, so what this means is, so there is something you call in vivo, which is inside the body. Inside the body, when things are inside the body, they are in their purest form, they are in their native form, and they perform the best, yes? But when things are outside of the body, they might be misfolded, so they might not perform optimally. So the reason, one of the reasons, you pointed to the reason, to that reason implicitly. The reason why we choose to go with the cyborg formulation is biology is ultimately very powerful. Yes? So a cell is a factory. It's capable of making its own proteins. It's capable of keeping its own proteins bound to the surface. And eventually, a protein that is on the surface of a cell that is properly folded is much more efficient. Therefore, when we are able to replicate this array that I talked about in a very, very large scale, we can capture far more VOCs that are coming in a non-invasive way as compared to our currently existing methods. So what we are imagining is not a system where you breathe on a device and somebody has to take you to a hospital and then you analyze it in the hospital and you get your result back. The future we imagine for healthcare is this. You come into your bathroom, you breathe on our device, within 10 seconds, we tell you if you have an illness or your state of health or your gut microbiome is doing, what have you. Yes? You get an instant recommendation of what you want to do about it, right? We want to make the patient the CEO of their health, if you will. But baby steps. Right now, we're at a stage where we have a capability to manufacture 50 of these devices. By the end of this year, we want to be able to make thousands of these devices and begin clinical trials in this year as well.
So, this is really interesting. Just one number. Right now, how long does the cell survive on a chip?
So right now, our chips survive for 1 month.
What will you change that it survives longer? And what is the target price for a chip in mass production?
So we have to engineer the cells a little bit better, right? So right now, most of the engineering or most of the genetic modification we have done to the cells is to make them more specific and sensitive. What does that mean? We want cells to respond faster. We don't want you to have to wait 30 minutes. We don't want you to have to wait for 10 minutes. We live in a world of do it now, right? So we have mostly focused on this. And right now, there is not a strong business reason to keep the cells alive longer. But over the next years, one of the metrics of how our products perform, at least internally, is to be able to extend the cell lifespan for a year, 2 years. We have demonstrated in the lab that we can keep cells alive for 1 year. Okay. In terms of the target price, we are targeting the global middle class. On this planet today, there are 2 billion people that have never seen a doctor, like ever. In rural America, 80% of the people are in a place where they are disadvantaged health-wise. Yes, and in China, for instance, to get access to healthcare, the same way that people will get access to healthcare here in Switzerland, the government of that country will have to increase healthcare spending by 700%, right? And that situation is not going to improve either in the developed world or in the so-called developing world. So we have to do something about healthcare. So for us, we are targeting that global middle class that can afford a mobile phone. Because the situation where 90% of the reasons why people go into hospitals, you can essentially analyze their disease with a sensor and be able to make some automated decision out of it, would take away a lot of load from the doctor class you have so they can focus on more intelligent things.
Thank you. Some questions from the audience? No questions? Okay, us. Thank you very much for the interview.